If you've seen any sci-fi movies like Vanilla Sky or TV shows like Futurama, you might be aware of its depiction of cryopreservation, a process that involves completely freezing biological samples. These samples can range between microbes, animals, and maybe even humans. And in the case of the University of Sao Paulo in Brazil, their samples were horse semen. Renata Lensoni and her colleagues looked to understand stallion spermatozoa, or sperm cells, and particularly the cellular process of ubiquitination. Put simply, tagging a substrate for its inevitable destruction. Sperm cells that don't move properly or look a certain way will be tagged with ubiquitin. This tagging is part of the ubiquitin proteasome system, or UPS for short, a signaling pathway found in the nuclei of sperm cells. Xiaonan Zhang and her colleagues from the University of Minnesota describes this perfectly. First begins step 1 activation. A free-floating ubiquitin molecule is activated by attaching to an E1 ubiquitin activating enzyme via ATP phosphorylation. Step 2 is conjugation. The activated ubiquitin is transferred onto another molecule called the E2 ubiquitin conjugating enzyme. E2 works like a taxi, transferring the ubiquitin to its final destination, where step 3, ligation, occurs. E2 deposits the protein to the E3 ubiquitin ligase enzyme, a complex that connects the ubiquitin to a targeted substrate, in this case, parts of a sperm cell. Steps 1 through 3 continue to occur until the targeted substrate is bound with plenty of ubiquitin, in which the fourth step, degradation, occurs. The 26S proteasome senses signals of plentiful ubiquitin on the substrate, where it works to break it down into single peptides, and the ubiquitin is recycled anew. This is protein degradation via ubiquitination, the main signaling pathway studied in this paper. So what did Lencioni and her colleagues do exactly? As mentioned before, the overall purpose of this paper was to understand protein degradation in horse sperm cells that both fail to freeze, cells with poor freezability, aka the PF group, or succeed in freezing, cells with good freezability, aka the GF group. They hypothesized that samples in the GF group will demonstrate lower levels of ubiquitination, protein degradation, and perhaps morphological defects in comparison to their PF counterparts. We can summarize 3 overarching outcomes of their results. In order, they include the differences in sperm motility, the differences in morphological defects, and the differences in sperm ubiquitination intensity. The first was motility. In one paragraph, the authors list average percentages of post-thaw progressive motility, or the amount of sperm movement after thawing from cryopreservation. They reported that PF stallions had limited motility at around 31.3% compared to their GF stallions with a whopping 73.7%. The following table is a statistical t-test with a 95% confidence interval showing the percentage of sperm defects observed in both groups. Clearly, there is a higher rate of defects in PF groups in comparison to the GF groups, with the only exception being the proximal and distal droplets, but only marginally. Now take a look at another t-test figure. The following graph depicts the percentage of ubiquitin in PF samples in dark gray and GF samples in light gray, showing how great the ubiquitination intensities in PFs outweigh the other. This micrograph is an immunofluorescent staining in both PF and GF samples. Panels A, B, and C increase in primary antibody staining in green, solely illuminating ubiquitin, with panel D being a negative control. Panel A is from a GF sample, has little ubiquitin, and has a straight tail, whereas B and C are from PF samples, with more ubiquitin and are deforming. Recall that PF stallions had lower motility in the first finding, and this illustrates why. Lencioni and her colleagues demonstrated how sperm cells with poor freezability are likely to experience higher levels of protein degradation and therefore morphological defects. But whether or not a sperm cell freezes is dependent on how it was cryopreserved. Is the sample cold? Has it been cold long enough? Are there any other signaling pathways besides ubiquitination involved? What other secrets lie beneath the ice? It's clear to see that protein degradation is extensive, but simple to understand. Simple enough to understand that cryopreserving life is as interesting as ever, whether it's for sperm, human sperm, or Fry from Futurama.